My name is DJ. I'm the CEO of the ACRO Biomedical company. And it's a great honor for me to present our company here. ACRO Biomedical was founded in June 2014 and is located in Kaohsiung Science Park in Taiwan. And the registered capital is $13 million US. And we have received 43 international patents, and there are more than 20 other patents pending. And we received 3 FDA 510K, 5 TFDA Class 2 medical device, and 2 Class D from Singapore, Philippines, Vietnam FDA medical devices. And the company was IPO in July 2021 in Taipei Emerging Stock Exchange Market. And we received numerous awards during the past few years. And regenerative medicine is a fast-moving industry. The annual compound growth rate is 26.1%. The reason for this high, fast-moving industry is because, according to WHO, there are severe shortage of tissues and organs around the world. There are less than 10% of the organ transplantation has meet the market needs, and that means there are 90% more people are waiting in line. And this situation has caused the fast moving of regenerative medicines. So our mission is to use animal-derived biomaterials to regenerate human defect, tissues, and organs, and ultimately become the global leader in tissue engineering and regenerative medicine field. Our vision is that one day people do not need to wait in line for donated tissues and organs. Tissue and organ transplantation will become easily available and affordable. This is our platform technology. We use the proprietary supercritical CO2 extraction technology to remove the immunogen from animal tissues and organs and leave the natural collagen scaffold as biomaterial for tissue and organ regenerations in human. We consider this technology as the holy grail technology for the biomaterial preparations. As you can see here is a porcine skin with dots of cells. But after the supercritical CO2 extraction, all the cells has been completely removed. And under electron microscopy, you can see that the collagen scaffold remains intact. This is a minimum manipulated process. The cost is very low, and the process does not disrupt the natural collagen scaffold structure. And there is no chemical solvents involved in the process, and it can kill bacteria and virus at the same time. And it's an environmental friendly process. You can recycle the CO2. And it can be applied to various tissues and organs, also tissues and organs from various animals, including porcine, bovine, equine, and ovine, or also human tissues. And it drastically reduces the immune rejections. Here are some of the SEM structure of the cellular, like porcine dermis, cancerous bone, cartilage, tendon, cornea, blood vessel, nerve, ureter, and heart, liver, and kidney. And you can see that the alignment of the collagens and also the mechanical strengths varies amongst tissue to tissues and organs to organs. So our ultimate goal is to use the biomaterial from animal tissues and organs to fix human defects from head to toes, from outer layer to inner organs. Here are our product pipelines. We have Class 1 medical device, Class 2, Class 3 medical device, and also Class 3 medical device. We also have product involved in stem cells for skin cell therapy and knee joint cell therapy. And we also work on organ regenerations. And the market size of the product line we develop are huge in the global market and also in Taiwan and China markets. And we have product for tissue engineering and aesthetic medicine, pet health care, and also biomaterial for research use only. And here are the milestone activities started from the establishment of the company up until now. And here are the patent portfolio. Our patents cover the corneal transplantation, cartilage regeneration, aesthetic microsurgery, skin slicing, corneal slicing, and also organ regenerations, bone regeneration, and hair regenerations. And the red dots represent the patent granted, and black dot represent pending patents. And currently, we receive 43 different patents from different countries. And let's see the porcine derived product with this decellularized dermal collagen matrix powder, and a collagen membrane for dental applications. And we also have this dermal filler for wrinkle injection. And this is the ACRO Collagen Solutions, which is registered in US FDA as device method file number. And also we have these skincare, wound care products to prevent scar formations. We have a dress and spray, gel, and aesthetic processes. And we also have these skincare series of products. And the collagen matrix are intended for all kinds of wounds, including burn wounds, diabetic ulcer wounds, traumatic wounds, and surgical wounds. And it also has many off-label use. And we have received US FDA, Taiwan, Singapore, Philippines, Vietnam FDA approval. And you have various shape and sizes. And the dental membrane for dental GTR and GBR operations, and it's come from the cellular dermal matrix, and it's got Taiwan FDA approval. And the porcine bone derived, we grind the porcine bone into granule, cut them into cube, stripe, or wedged form, and these plug form. And we also have this Lego form of the bone. You can put all the bones together to form a lower jaw. And this is intended for oral cancer patient. When you have a large maxillofacial defect, we can fix it at one time, one surgery. And we also have the bone booty and the cartilage graft for knee joint regenerations. So you can see those bone material for orthopedic and dental applications. And for orthopedic, we have received US FDA, Taiwan, Singapore, Philippines, Vietnam FDA approval. And for dental application, Taiwan has approved. And we also have these various shape and size of the bones matrix, and got US FDA and Taiwan FDA approval. And cornea, this is the corneal substitute, decellularized from porcine eyeballs. We purchase the eyeball and remove the cornea and go through our decellularization process, and it becomes a porcine decellularized cornea, which is ready for corneal transplantation. We first work on pet dog Bonnie. Bonnie's right eye suffered from cataract and corneal scar, so the operation is simple. We simply just remove the cornea and then replace the intraocular lens, and on top of that, we put the porcine decellularized cornea. And 1 month later, the right eye becomes clear and visible. This is the first canine corneal transplantation in the world. And currently, this corneal product is under human clinical trial in 40 medical center in Taiwan. Hopefully, we can get this clinical trial done this year and get the PFDA approval. And this is dermal filler. It's a world's first particle form of dermal filler. The particle come from this decellularized dermal matrix. We cryo-grind it into particles and put the various size of the particle together with the collagen fiber slurry into this syringe, and it's ready for wrinkle injection. And because it's particle form, each particle will consist of this natural collagen scaffold. So the skin stem cells and fibroblasts can come into this scaffold to proliferate and secrete more extracellular matrix. So as compared to those fiber form of collagen dermal filler, the particle form will last much longer under the skin. And we expect this product to last more than 12 months under the skin, and it will outcompete the current product with 3 months duration and 6 months durations. And our research also find that when you put this cellular dermal matrix particle under the animal skin, not only do we see a new collagen formations, we also see new blood vessels and new hair follicle. This hair follicle regeneration makes us so excited, so we write a new patent for this bald head market. This is the histology. When you inject the cellular dermal matrix particle under the rabbit skin, and 30 days later, you see a lot of new collagens and also new hair follicle and new blood vessel. As comparison, we do not see the same effect by sculpture and wrestling. We also perform this experiment on new mice, and new mice basically does not grow healthy hair. But when we inject those cellular dermal matrix particle under the skin, you can see that 30 days later, a lot of hair actually come out of the skin. And this is skin cell therapy. We combine the natural collagen scaffold together with human mesenchymal stem cells, and to use this to regenerate the skin. And you can see that the regenerated skin has epidermal and dermal layer and also hair follicle. And by this method, we can reconstruct the whole skin within 18 days. And we also do the knee joint cell therapy. We first grind the knee joint cartilage into granule and then decellularize by our technology. And we put those decellularized cartilage graft together with PRP and chondrocyte, put it into these defected areas. And 30 days later, we check on histology. And you can see that the whole defect area has been reconstructed with the cartilage. And if you enlarge this area, you can see that each lacuna has got chondrocyte inside. And if you look closer, this is the original cartilage. This is the graft material we put in. And they look exactly identical within 30 days. So we can actually reconstruct the knee joint defect within 30 days. And our ultimate goal is to do organ engineering in vivo. So we decellularize heart, kidney, and liver, and put those decellularized organ scaffold back into live animal and connect the blood circulation so that the blood flow will bring in all the stem cells to reconstruct the organs in vivo. After it is fully reconstructed and functional, it will be ready for autologous organ transplantations. So to prove the concept, we did a rabbit model. We decellularized rabbit hearts and give it to the heart surgeon. They put the decellularized rabbit heart back into another rabbit, put it in the abdominal cavity, connect the blood circulations, and you can see the blood flow coming to this heart scaffold, and then close the wound and wait for the organ to be reconstructed inside the rabbit. And if it works, this rabbit will have 2 hearts pumping at the same time. The same idea is applied to kidney reconstructions. We get a rabbit and a healthy rabbit, remove one of the 2 kidney, and then put this decellularized rabbit kidney on the spot and connect the blood circulations. And you can see the blood flow coming to this decellularized kidney scaffold, and then close the wound and wait until fully reconstructions. And these are all underway, still working on it. And we also have this scaffold material for research use. We decellularize all kinds of tissue and organs and put them into a petri dish, so scientists can culture stem cells or somatic cells for tissue engineering study. And we also grind those scaffold into powder so that scientists can use those powder for 3D bioprinting. They can print any tissues and organs using those materials. And here are some of the publications we published in the past 2 years, and trademark, brand name, and logos registered all over the world. And some certificate, including the ACRO Collagen material, FDA device master file, and also class 1 medical device, Taiwan and European community, and also US. And these are US 510K and Taiwan FDA class 2 medical device. And Singapore, Philippines, Vietnam, collagen matrix, and bone graft. And these are the awards we received in the past few years. And this is Discovery Channel came to feature Bonnie's story. This is Dr. Chai performed the corneal transplantation for Bonnie, and these 2 are Bonnie's hosts. And you can see the link in YouTube. And Fortune Investor Guy actually featured our company story. The title is 2 Hearts Are Better Than One. It's last year, a report. And we also signed contract with Australian company, OBM, and Japanese company, Marubeni, and also Tri-Service General Hospital. And we have media release. And we were invited to give speech all over the world. This is Hong Kong, this is Kyoto, and this is Johns Hopkins University in Maryland, and Dalian, Xi'an, and also Suzhou, and Philadelphia Temple University, and also Taiwan Defense Medical Centers, and Hangzhou, and also Bio US. And our slogan, we continue to make the impossible possible. Thank you very much for listening, and hope that we will have the chance to meet each other in person and to discuss more collaboration projects. Thank you.